Welcome back to the Science Experience, where we explore the most exciting new research papers and preprints just for you. One of the biggest puzzles in understanding how our bodies work is figuring out what happens inside our brains as we age.
Yeah, it's incredibly tricky.
It's not like we can easily, peek into living human brain cells over decades.
Not ethically, anyway.
Right. But what if we could grow those very brain cells in the lab functioning neurons and actually watch the aging process unfold?
It sounds futuristic, but it's really happening.
It might sound like something out of a sci-fi movie, but it's becoming a powerful tool for scientists, and that's what we're diving deep into today.
Absolutely. The ability to create models of human aging in a dish is, well, it's a game changer. We can start to unravel the fundamental biological mechanisms. Today, we're focusing on a really compelling study that asks a crucial question. How accurately can these lab-grown neurons replicate the aging we actually see in the human brain?
Exactly. The paper we're unpacking comes from the Nature Portfolio journal Communications Biology. It's titled Tracing Mitochondrial Marks of Neuronal Aging in iPSCs-derived Neurons and Directly Converted Neurons. And the brilliant minds behind it are Nimi Varghese, Linora Sabo, M. Zamil Kader, Imani Lejri, Amandine Grimm, and Ann Eckert.
Quite a team.
Now, for you, the learner understanding how our brain cells age is so important. It's at the heart of everything, from those, tip of the tongue moments
You've all been there.
to the development of serious age-related neurological conditions. This research gives us a really innovative way to tackle these questions without getting lost in overly complex biology.
Right. Our mission in this deep dive is to extract the core insights from this study. We specifically want to understand how two different types of lab-grown neurons, those derived from induced pluripotent stem cells, or iPSCs, and those directly converted from other adult cells, how they reflect the aging process. And a major focus here is on the mitochondria. As you know, the tiny powerhouses within our cells generating energy, how these energy factors are affected by time, it's absolutely critical.
Okay, let's unpack this. So, studying aging in the human brain directly, it presents some pretty significant hurdles. Huge hurdles.
Definitely.
But this study looks at two fascinating workarounds. iPSC-derived neurons, or iPSCsNs, and directly converted neurons, or INs. Can you break down what these are and why they're such exciting tools?
Certainly. Both iPSCsNs and INs are novel in vitro models. They let researchers study human neurons outside of the living body. The basic idea is taking adult cells like skin cells, they use fibroblasts in the study, and essentially reprogramming them to become neurons.
Right, reprogramming.
But the way this reprogramming happens, that's where the key difference lies.
And that difference is really important, right? Because I remember reading that iPSCsNs go through this stem cell-like phase. What does that actually mean for their age?
Precisely. When cells are reprogrammed to become iPSCs, they essentially revert to a state very similar to embryonic stem cells.
Like hitting reset.
Think of it exactly like a biological reset button. This process can wipe away some of the age-related changes that were present in the original adult cell.
Okay.
On the other hand, directly converted neurons, or INNs, they're thought to skip this full stem cell-like phase.
So they don't get fully reset.
The idea is that they hold on to more of the aging characteristics, the hallmarks of aging, from the original donor cell.
So INS might be like a snapshot of the older cell's age, while iPSCsNs might be a bit more youthful after that reset. That makes sense.
That's the working hypothesis, yeah.
Now, what about the practical side? Growing these two types of neurons, are there big differences in, how long it takes or the cost?
Yes, labs definitely have to consider these things. Generally, creating iPSCsNs takes longer. That intermediate stem cell stage adds time.
Right.
However, once you have those iPSCs established, they can be grown pretty much indefinitely. So you get a large, consistent supply of cells.
Useful for big experiments.
Exactly. Really useful for big experiments or drug testing. In contrast, making INs is usually faster, maybe fewer weeks.
Quicker turnaround.
But you're limited by the initial number of skin cells you started with, which can make large-scale studies more challenging.
Right, so iPSCsNs are like building a factory takes time, but high output. INs are more like a custom workshop, faster start, limited output.
Okay.
That's a good analogy.
So the study really hones in on mitochondria. Why are these tiny parts of the cell so crucial when we're talking about aging?
Mitochondria are just fundamental for life. Their main job is producing energy, ATP, the cell's fuel,
The powerhouse.
the classic powerhouse, yes.
Yeah.
But they're also involved in many other vital processes. What's particularly interesting and relevant here is that mitochondria are quite vulnerable to the effects of aging. Over time, their function can decline. This leads to energy problems, oxidative stress, other issues within the cell.
And neurons need a lot of energy, right? Exactly. Neurons have very high energy demands. Their reliance on healthy mitochondria makes them especially susceptible to age-related mitochondrial problems.
So this study took a really close look at different aspects of mitochondrial health in both types of lab-grown neurons, the iPSCsNs and the INs, and compared them to each other, and also back to the original skin cells, the fibroblasts, from both younger and older individuals.
A very thorough comparison.
Let's start with those original skin cells.
Yeah.
What signs of aging were actually present in the fibroblasts from the older donors?
The researchers observed several telltale signs of aging in those older fibroblasts. They found lower levels of ATP.
Less energy.
Less energy production, yes. The mitochondrial membrane potential you can think of, that like the charge in the cell's tiny energy batteries, it's crucial for generating power, that was also lower.
Okay.
These aged fibroblasts also showed a slower rate of respiration, meaning their mitochondria weren't using oxygen as efficiently to produce energy.
Less efficient power generation.
Correct. And interestingly, they also saw a drop in the NAD plus NADH ratio. Think of NAD plus as a key helper molecule for energy production. When it's used, it becomes NADH. A lower ratio often indicates metabolic strain or stress.
Oh, yeah.
On the flip side, they found more mitochondrial free radicals, those unstable molecules that can damage cells.
The bad guys.
Sort of, yeah. And an increase in the overall number or mass of mitochondria, possibly as a way to compensate.
Trying to make up for the inefficiency.
Potentially. And finally, the network of mitochondria within the older fibroblasts appeared more broken up, fragmented, rather than a healthy, interconnected web.
That's quite a list of changes. It really sounds like time impacts the powerhouses of these skin cells significantly.
It does?
Now, what happened when they transformed these older fibroblasts into directly converted neurons, the INs? Did these neurons show similar aging characteristics?
In many ways, yes. The aged INs did mirror the aging features of the original fibroblasts. They also had lower ATP levels and a reduced mitochondrial membrane potential.
Consistent there.
Yes. There was a trend towards more free radicals, although it wasn't statistically significant across all measures they looked at. Interestingly, though, they did see a significant increase in an antioxidant enzyme called CAT.
Trying to fight back against the free radicals.
Perhaps, yeah, maybe a defense mechanism kicking in. Importantly, the aged INs showed a significant decline in their overall mitochondrial bioenergetic capacity. Their ability to generate energy through respiration was clearly reduced.
So less efficient again.
Right. And, just like the fibroblasts, they showed a notable increase in glycolysis.
Ah, the backup energy pathway.
Exactly. Another way cells can produce energy, suggesting they were shifting towards this alternative pathway, maybe because the main mitochondrial one wasn't working as well.
Makes sense.
The aged INs also had that lower NAD plus NADH ratio and a more fragmented mitochondrial network. Plus, they saw increased activity in genes involved in both mitochondrial fusion joining together and fission splitting apart.
So the network dynamics were affected too.
Yes, and also a tendency for more mitochondria overall. Again, similar to the fibroblasts.
So it sounds like the direct conversion process really does allow these neurons to hold on to quite a few of those aging hallmarks, particularly around energy production.
That seems to be the case.
So if you're interested in studying how aging impacts a neuron's ability to generate power, ions seem like a really relevant model.
That's a key insight, I think. The aged INs appear to be a valuable in vitro model for specifically studying how aging affects a neuron's energy production machinery.
Now let's switch over to the iPSC-derived neurons, the iPSCsNs. Remember, they went through that stem cell-like reset. Did they still show signs of mitochondrial aging?
Yes, the aged iPSCsNs did show some trends consistent with mitochondrial aging. They also showed trends of decreased ATP and mitochondrial membrane potential.
Okay, similar so far.
Aligning with what was seen in fibroblasts and INs. They also had more mitochondrial free radicals and a significant increase in another antioxidant enzyme, GPX1 this time.
Another defense enzyme.
Correct. Moreover, they exhibited a significant decrease in mitochondrial respiration.
So, again, the core energy production was down.
Yes.
Okay, so some similarities there. But I recall something interesting about glycolysis being different in these iPSCsNs.
Right.
What did they discover?
This is a really crucial distinction. Unlike the aged fibroblasts and the aged INs, both of which showed an increase in glycolysis,
Right, the backup system.
the aged iPSCsNs displayed a substantial decline in glycolysis.
Wow. Okay. So the opposite reaction.
Completely the opposite. This suggests that while they experienced mitochondrial impairments, their overall energy metabolism responded to aging in a fundamentally different way compared to the INs and the original fibroblasts.
Fascinating.
Additionally, the aged iPSCsNs showed a significant decrease in the level of NAD+ and an increase in NADH, leading to that lower NAD plus NADH ratio. They also had a fragmented mitochondrial network.
Still fragmented.
Still fragmented. Similar to the other aged cell types, they had increased mitochondrial mass. But interestingly, there weren't significant changes in the genes related to mitochondrial dynamics, fusion, and fission in the iPSCsNs.
Another difference from the INs.
Yes.
That's a really interesting divergence, especially with the glycolysis.
Yeah.
It makes you wonder what that stem cell phase does to sort of rewire their metabolic responses to aging.
It's a key question.
So when the researchers compared these two neuron models directly, what were some of the key differences that really stood out in, say, their gene expression?
Well, they used a technique called principal component analysis.
Like a way to see the big picture in the data?
Exactly. It helps visualize the biggest overall patterns in all the genetic data. This analysis revealed significant differences in mRNA expression between the aged INs and the aged iPSCsNs. It indicated that even though they originated from the same aged donor cells, their genetic activity really diverged after the different reprogramming methods.
Okay. Any specific examples?
Yeah, for instance, while both models showed increased expression of NRF2, which is like a master control switch for keeping mitochondria healthy with age.
So that was consistent.
The upstream regulators of PGC1, another important player in mitochondrial health and creation, showed contrasting patterns between the two types of neurons. And genes like PRP1 and UCP2, which were more active in aged INs, and that's consistent with aging in other tissues, were not significantly changed in aged iPSCsNs.
More evidence of divergence.
Definitely.
And there was also this visual spiderweb comparison. Looking at individual donors, what did that tell them?
Ah, yes, the spiderweb plots. They compared various mitochondrial parameters across the different donors and their different cell types, fibroblasts, INs, and iPSCsNs. They really highlighted how individual the aging process can be. But interestingly, they found more similarities among the aged donors across their different cell states compared to the younger donors.
So older individuals looked more alike in their cell profiles, regardless of cell type.
It suggests that the aging process itself might push cells towards a more common functional state, maybe a less optimal one, regardless of their starting point or how they were reprogrammed. It emphasizes the individual nature, but also perhaps a common endpoint of aging.
Fascinating. Now, one of the, classic signs of aging is the shortening of telomeres, those protective caps on the ends of our DNA. What did the study find about telomere length?
Right, telomeres. As expected, the researchers observed telomere shortening with age in the original fibroblasts. That fits the standard model.
No surprises there.
But, and this is a really key finding, they found an increase in telomere length after reprogramming fibroblasts into iPSCs, regardless of the donor's age.
An increase, so they got longer.
Yes. It's almost like these cells got a fresh start on the telomere front during that iPSC stage.
Wow.
It strongly suggests a rejuvenating effect on telomeres during that pluripotent stem cell stage.
Okay, that's big.
But interestingly, the telomere length and also the activity of telomerase, the enzyme that maintains telomeres, did not significantly differ between young and aged iPSCs themselves, or between the young and aged INs, or the iPSCsNs.
So the difference showed up in becoming iPSCs, but not later between young and old neurons.
Exactly. The researchers suggest that maybe mature neurons, which don't divide frequently, might not rely heavily on telomerase activity anyway, so that initial reset doesn't translate into ongoing differences in the neurons themselves based on donor age.
Interesting nuance. Okay, so bringing it all together, what were the overall conclusions of this study? What are the big takeaways for us?
The study concluded that both aged INs and aged iPSCsNs show clear signs of impaired mitochondrial energy production. This supports the well-established idea that oxidative stress and mitochondrial dysfunction are major players in aging.
Okay, that's confirmed.
But the critical difference lies in their metabolic response. Aged INs seem to rely more on glycolysis, that backup energy source, while aged iPSCsNs do not. They actually decrease it.
The key divergence point.
Precisely. Based on the broader range of retained aging features, especially the metabolic similarities to the original fibroblasts, the researchers suggest that INs might be a more accurate overall model for studying neuronal aging.
But iPSCsNs aren't useless.
Not at all. They emphasize that iPSCsNs can still be incredibly valuable for studying specific aspects of mitochondrial neuronal aging, particularly because they're generally easier to produce in the large quantities needed for many experiments.
Right, the supply issue.
Yeah. The study did acknowledge some limitations, like difficulty in perfectly matching genders for all comparisons, because generating INs can be technically challenging.
Understandable.
But they noted that previous research suggests gender has minimal impact on the specific mitochondrial parameters they were investigating.
So, probably not a major confounder here.
Likely not.
So it sounds like both models offer unique advantages, unique windows into understanding how our brain cells age. It's like having two different lenses to view this really complex process.
Exactly. It's not about one being good and one bad, but about understanding their different strengths. This research offers valuable new insights into how these lab-grown neurons can help us break down the complexities of brain aging. It's a definite step forward in developing these in vitro models that could potentially be used down the line to study age-related diseases, maybe even lead to more personalized medicine approaches.
But still more work needed.
Oh, absolutely. The authors rightly point out that more research is needed to fully characterize these models and understand exactly how well they reflect what's happening in the actual aging human brain.
To recap then, we've seen that both types of lab-grown neurons from older individuals show mitochondrial signs of aging, things like reduced energy production, more oxidative stress.
Consistent findings there.
But the really fascinating part is how they handle their energy metabolism overall. The INs ramp up glycolysis, the backup system, while the iPSCsNs surprisingly do the opposite, they decrease it.
That metabolic difference is key.
It suggests that the journey a cell takes to become a neuron, direct conversion, versus that stem cell state, really influences how it responds to the wear and tear of time.
It really does. And this research provides, I think, a crucial step towards understanding these fundamental processes of neuronal aging. It gives us that aha moment, seeing both the expected similarities but also these surprising differences. And this knowledge could ultimately lead to better ways to maintain brain health as we get older.
And that brings us to a really interesting question for you, the learner. Considering these findings, what specific aspects of neuronal aging do you think would be best investigated using each of these models? You know, the directly converted neurons versus the iPSC-derived neurons. What kind of questions about the aging brain do you think each model could help us answer most effectively? Something to think about.
Definitely. Ultimately, this study highlights the exciting potential of using these advanced cellular models to unlock the many mysteries that still surround the aging brain. It's a rapidly advancing field, and these kinds of detailed comparative investigations are essential for moving towards a deeper understanding of how we age and, importantly, how we might be able to age healthier.
Absolutely. If you enjoyed this deep dive into the latest science, please remember to like the video and subscribe to the SANS Science Experience.
Yeah, your support really does help the channel grow.